Dear students, welcome in science class. Class 10, Unit 18B, Artificial propagation in plant. Topics: layering, simple layering, compound layering, tip layering, mound layering, air layering, grafting, whip grafting, tongue grafting, cleft grafting, tissue culture. Method of tissue culture. Artificial propagation of plant. The production of number of plants by artificial method is called artificial propagation of plant. Method of artificial propagation of plants are layering, grafting, tissue culture. Layering. The process of preparing new plants by growing root from the branch of parent plant is called layering. Layering are of 5 types: simple layering, compound layering, tip layering, mound layering, air layering, simple layering. The layering in which a branch is bent and inserted inside soil for 2 or 3 months to develop the new root and separated from parent plant is called simple layering. Compound layering. The layering in which a branch is bent and inserted inside soil at many parts for 2 or 3 months to develop the new roots and each part is separated from parent plant is called compound layering. Tip layering. The layering in which the tip of branch is inserted inside soil for 2 or 3 months to develop the new roots and separated from parent plant is called tip layering. Mound or stool layering. The layering in which the branches developed at base are inserted inside soil for a year to develop roots and separated from parent plant is called mound layering. Ear layering. The layering in which the external bark of a branch is cut out in form of ring and the wet soil is kept and tied with plastic for 2 months to grow roots and separated from parent plant is called ear layering. Grafting. The technique of transplanting a branch of one plant to another plant of closely related species is called grafting. Types of grafting are whip grafting, tongue grafting, cleft grafting. Sign. The upper part of grafting plant is called sign. Stalk. The lower part of grafting plant which is with the root system is called stalk. Whip grafting. The grafting in which the slanted cut scion and stalk are joined and wrapped with plastic making airtight and tied is called whip grafting. Tongue grafting. The grafting in which the slanted cut scion and stalk with tongue are joined and wrapped with plastic making airtight and tied is called tongue grafting. Cleft grafting. The grafting in which the wedge-like cut sign is inserted into cleft of stalk and wrapped with plastic, making airtight, and tied is called cleft grafting. Tissue culture. Tissue culture is modern technique of reproduction in which cell or tissue is cultured in sterilized and nourished medium to produce new plant in large number. Advantages of tissue culture. Large number of genetically identical plants can be produced. Rare and endangered species can be preserved. Sterile hybrid plant can be produced in large number. Process. Plant tissue is scraped from parent plant. Tissue samples are placed in sterilized medium with nutrients, cytokinin, and auxin hormone. The cells in the sterilized medium divides and forms into mass of cells is called callus. The callus develop into tiny plantlets. Plantlets are planted into compost medium. Importance of artificial propagation of plant. It can produce large number of plant in short period of time. It can reproduce the plants lacking seed or viable seeds. It can preserve endangered species of plants. It is easy and fast process of reproduction. It can produce genetically identical plants. It produces plants by asexual process. It produces fruits in short period of time. The sign and stock selected must be closely related species and Cambrian layer must be appropriately matched. Simple layering is done in lemon. Compound layering in apple, pear, and walnut. Deep layering in blackberry and raspberry. mound layering in apple, pear, plum, and mango. Auxin hormone develops roots of plant. Cytokinin hormone develops stem of plant.